afternoon, everyone, and thank you, Garvan College, for having me here. And the work that I'm going to talk about today is the study that I have carried out in my PhD from the Department of Biotechnology, IIT Kharagpur. The topic is development and characterization of osteogenic microenvironment mimicking scaffold for bone tissue engineering. Now, from the title itself, you can understand that my topic here is related to a tissue engineering field. So before I say anything about the study or my research, I will try to briefly talk about the history of tissue engineering, how it came to be one of the major fields in biomedical engineering, what different types of tissue engineering approaches are available, and how their limitation led to my research topic. The idea of tissue engineering emerged just sorry yes, the idea of tissue engineering emerged just over 30 years ago in 1988. The two men that were credited with doing this seminal work in this field are Joseph Vacanti, a surgeon, and Robert Langer, a professor at MIT. They published a paper in Science where they defined tissue engineering as an interdisciplinary field that applies the application or principles of both engineering and life science towards the development of a substitute or biological substitute that can restore, maintain, or regenerate functional tissue. However, at that point of time, the tissue engineering was not a very mainstream buzzword until almost a decade later when the infamous Vacanti mouse was revealed. Yes, it's a mouse with a human ear on its back. The mouse evoked quite backlash from animal rights activists and many other organizations. But for many others, the reaction was one of the excitement and wonder. I mean, they thought that if we can grow a human ear, why not a kidney, a liver, or an eye? And thus emerged an era of tissue engineering filled with hope and hype. So the Vacanti mouse is still impressive to this day and represented a major step forward in science. If you can see this graph, right after the revolution of Vacanti mouse around 1998 and 2000, the publication in field of tissue engineering has been skyrocketing. So when I'm talking about tissue engineering, I already told about the definition of the tissue engineering. So what is this process of tissue engineering? How we prepare a three-dimensional construct or a scaffold that can develop into an organ or tissue. So what we do is generally we collect cells from patients or animals, and these cells were then cultured in lab, expanded in greater number. Then a biodegradable material is fabricated, formed into a shape of a target organ or tissue. And these biomaterials are then seeded with the cells cultured in the lab, and then they were implanted or in the patients after maturation. And it was hoped that the scaffold will slowly degrade away as it as the cells proliferate, and it forms or reconstruct a functional organ or tissue. Now, in case of a Vacanti mouse, just before the slide, the ear here was actually a mesh of biodegradable plastic that was molded into a desired shape, and they were sprinkled with some cartilage cells collected from a cow and implanted under the skin of mouse. The researchers removed this ear after 12 weeks and found that some new cartilage has been generated within the structure. So, however, despite these 30 years of dedicated research in tissue engineering, we are still relying on organ donor rather than growing kidneys, livers, and hearts in the lab. In this regard, I have looked into the development of hard tissue or skeletal tissue and in skeletal tissue engineering. However, before moving into my research into skeletal tissue engineering, let's look into what is the current scenario of orthopedic trauma management throughout the world. So according to World Health Organization, over the past few decades, musculoskeletal disease and injuries kills more lives than HIV/AIDS, malaria, and tuberculosis combined. And this affects more in lower and middle income countries. Over next 15 years, it was estimated that almost close to 8 trillion of cumulative GDP will lost due to a decrease in economic productivity because of this orthopedic disease and trauma. And to treat these orthopedic problems, most of the patients still opt for grafting procedures, that is either autograft or allograft. Autograft is graft within the same individual, or allograft is within the same species. Now, among these graft procedures, autologous graft or autograft surgery is still considered the gold standard. But there are problems with autografts, such as shortage of available grafts, site morbidity, there are chances of infections or immune rejections. The surgical procedure is quite laborious and high cost of graft surgery. So all these problems have caused clinicians and scientists to look towards an alternate approach. And in this context, many scientists and clinicians believed and considered that bone tissue engineering has a tremendous opportunity as an efficient and a permanent solution, where the scaffold and cells are used in combination to replace or regenerate the disease part. So the whole tissue engineering method or approach can be broadly classified into two types. The first one is a classical or a very traditional approach I already mentioned in my previous slides. We call it top-down approach, where we add cells in a three-dimensional structure or scaffold, culture this scaffold in the lab so that the cells can grow and proliferate, followed by implantation into the patient. Now, this top-down approach has some potential in engineering tissues with a less complexity, such as skin. However, this approach has often failed to generate proper outcome with a higher complexity of tissue structure. Now, because of such persisting problems with this top-down approach, the conventional, researchers tried to find an alternate approach, and they came up with a method called modular approach or bottom-up approach, where it focuses on creating some microstructural or functional tissue modules. These tissue modules were then placed close to each other, allowed to fuse together to form the engineered tissue construct. Researchers found that this approach was able to mimic the complex tissue architecture, but problem is, since we are not using any 3D solid scaffold here, the application of this approach is only limited to soft tissue engineering and not suitable for hard or skeletal tissue. Further, since we are here using different tissue modules, they cause a necrotic or dead cells core that bring down the efficiency of tissue construct. So in this regard, to create an engineered bone tissue, we have come down with a combined approach. An approach with combination of both top-down and bottom-up methods. We hypothesized that if we seeded these tissue modules non-compactly on three-dimensional scaffold or matrix, this combination may have an efficient tissue regeneration almost similar to autogenous graft. However, this problem. What is the problem? Unlike single cells, which has a dimension around 20 to 30 micrometer, the tissue modules are bigger in dimensions. They reaches up to 200 micrometer in size. And so therefore, to non-compactly seed these tissue modules into the scaffold, we have to change the architecture of the scaffold. That is, to increase the pore size or porosity of the scaffold to make it more macroporous in nature. In most of the bone tissue engineering, the 3D scaffold that are made are from synthetic materials or polymers, such as ceramic, polyurethane, glass bead, polylysine, etc. So, and they use this synthetic material because they are very mechanically strong and stable. However, problem is the fabrication method for this synthetic biomaterial allows them with a very small pore size and porosity. The other disadvantage of this synthetic polymer is that they don't contain any kind of bioactive molecules, and therefore they doesn't allow cell adhesion and growth. So from here, I come to the first problem towards this combined approach is we have to fabricate a three-dimensional scaffold that has a macroporous structure or have macroporosity using natural polymers. So now there are very handful of techniques present, such as salt leaching, rapid prototyping, that can induce macroporosity. But again, these are all limited to synthetic polymers. So to induce macroporosity in natural polymers, we have used here a very simple and frugal method of high-speed stirring based foaming and then freeze drying method. We used natural polymers in these techniques is gelatin. This gelatin is a degraded product of collagen, and collagen, as you know, is one of the most abundant protein that present in the human body as well as in bone. So this gelatin solution is then mixed with our second component is called chitosan, which here acts as a thickener. The third component we have added is hydroxyapatite. This hydroxyapatite here added as an inorganic component and is a good source of calcium phosphate. So all these three components mixed together, and this solution was stirred at a very high speed of 1200 RPM, which generates foam or froth. And at this point, we stabilize this froth by cross-linking the polymer using butyraldehyde. And after stabilizing the froth, we keep this solution at minus 20 degrees Celsius overnight. And thereafter, we undergo for a freeze drying method for next 24 hours, resulting in the formation of a three-dimensional scaffold. As you can see from these two images, the left one is a scaffold with foam, whereas the right one is a scaffold without foam, only freeze drying method. So this scaffold was then imaged under scanning electron microscope, and the results revealed that an average of 600 micrometer size of pores formed in this macroporous scaffold. The dimension is almost 3 times to the pore size of a scaffold that was prepared just under freeze drying method, or the control scaffold. We have also measured the porosity using micro-CT scan. Porosity means the percentage of void volume in the scaffold. And here we found that approximately around 83% of the porosity in the macroporous scaffold as compared to the control scaffold, where it's only around 65% porosity. So this study proves that this combination of high-speed stirring based foaming and freeze drying method has been successful in fabricating a macroporous scaffold using natural polymers. In the next study, we determined whether this presence of big pore size or porosity in the macroporous scaffold was enough to allow the penetration of bone tissue modules for a successful combined approach. So for this, we prepare bone microtissue using a hanging drop method. This bone microtissue was measured of around approximately 187 micrometer. The microtissue was then placed on the scaffold, both macroporous scaffold and the normal control scaffold. And after a culture of 14 days, a micro-CT scan was done to determine how this bone microtissue modules has been formed in the scaffold. Now, based on the calculation, we found that the bone microtissue has been distributed to almost 90% of the volumetric area of the macroporous scaffold compared to only 42% area of the control scaffold. Next, we look into the second objective of this study, which is related to the biomechanics of the scaffold. Now, since it's obvious that since our study is related to the development of a scaffold or a construct for hard tissues, we have to develop a three-dimensional structure that should have an optimal mechanical strength comparable to the surrounding tissue. However, based on very previous data, if we increase the porosity of the scaffold, we lose the scaffold's modulus or the scaffold strength. So to solve this problem, we have thought of incorporating some other components. Here we have added nanofillers into these polymeric solutions to increase the mechanical strength of the scaffold. So we have used here 2 different nanofillers. The first one is multi-walled carbon nanotube. Many of you have heard probably about the carbon nanotube. It is widely used in many biomedical applications. due to its exceptional mechanical, optical, or electrical properties. The other class of nanofillers that has been used in this study is a type of dendrimer. We call it as a polyamide to amine, or short form is PAMAM, generation 4 PAMAM. Due to its molecular uniformity and multiple end functional groups, this molecule has been widely used in drug delivery studies. So, but these molecules has never been used in tissue engineering applications. So we fabricate the scaffold using this we explained same in our first objective. Here we have started with gelatin chitosan polymer, and then we mixed with either multi-walled carbon nanotube or with PAMAM. And after mixing mixing this component, we go for a high-speed stirring, stabilization of the foam, and then freeze dried to form the scaffold. And this scaffold were formed, then we tested its mechanical strength. We tested this mechanical strength using UTM, Universal Testing Machine, and another machine called nano indentation. So using UTM, we found that the carbon nanotube based scaffold shows a higher modulus compared to the PAMAM based scaffold. The modulus are almost 2.64 fold high for the carbon nanotube based scaffold and almost 1.92 fold high as for the PAMAM based scaffold compared to the macroporous scaffold. The nano indentation were used to determine the stiffness of the scaffold and were measured by a load displacement curve. Now we found that approximately 40% increase load is required for carbon nanotube based scaffold, and almost 20% increased load is required for PAMAM based scaffold for a similar displacement of 50 nanometer when compared to the macroporous scaffold. Now this study definitely indicates that there is an increase in the stiffness of the macroporous scaffold when we are adding a nanofiller. And moreover, the carbon nanotube incorporation shows a better, stiffer scaffold compared to the PAMAM based scaffold. However, this is not the end, because we are here adding since we are adding here the nanofiller into the scaffold composition, we are changing the chemistry here. And so what we want to know is whether this change in chemistry or changing composition of the scaffold is actually changing any biological related assay. That we wanted to determine whether there is any problems, any cytotoxicity happen in the when we are culturing with cells. So here we have conducted two studies. One is cell migration assay, one is angiogenic assay. So the first is the cell migration assay. It's a 2-day study where the study revealed that there's a higher cell migration happen in nanofiller based scaffold as compared to the macroporous scaffold. And among the nanofiller based scaffold, the PAMAM scaffold shows highest cell migration, almost 2.6 fold higher than the macroporous scaffold, whereas the carbon nanotube scaffold shows a almost 2-fold increase in migration. Further, we studied a chorioamniotic membrane assay, or short form CAM assay, to evaluate the pro-angiogenic nature of the scaffold. Now, in this assay, the material which is in question were placed on top of the membrane of a fertilized egg. This is what you see is a fertilized egg. This setup was then incubated for almost 7 days. The pro-angiogenic nature of the material was determined as by counting the number of blood vessels that is formed through or in and around of the material. And if you see here, we found that PAMAM scaffold shows highest number of microvessels or blood vessels, that which indicates its angiogenic nature. So from this biological study, we found that the PAMAM scaffold is comparatively better as compared to the carbon nanotube based scaffold. The reason we found is the PAMAM molecule contain large number of amine terminal groups. This positive amine terminal groups helps in cell attachments and blood vessel formation. So with this, we move forward in our next step, or last step, a in vivo study to determine the healing of a critical cranial defect. For this study, we have used Swiss albino mice, a gender female, aged 6 to 8 weeks. The material or the scaffold might create an immune reaction. So we have immunocompromised the mice using a mixture of cyclosporine or ketoconazole. Now, before the surgical implantation, these mice were anesthetized using diazepam and xylazine. So a 3 mm wide round defect was created using dental draining. Utmost caution was taken not to disturb the underlying durameter. The scaffold was then rinsed well in the sterilized buffer and placed gently on top of the scaffold. The skin was sutured and the animal was monitored till it regained sense. All the mice were then transferred and kept individually in an IVC facility. The daily administration, we daily administered the analgesic and antibiotic, including with semi-liquid food and water. And after 4 weeks of post-operative care, the mice were sacrificed by cervical dislocation. If you can see, the visual investigation of the sutured area reveals no sign of infection and allergic reaction. And after exposing the implant, we found that the size of the scaffold looks considerably reduced, which means that there's an in-vivo degradation of the scaffold happened. And a major part of the scaffold has, we found, attached itself to the defective area of the skull, which indicates an implant integration with the host tissue. To determine the extent of critical defect healing by the implanted scaffold, the calvaria was carefully extracted and was repeatedly washed with sterile buffer. And the 3-dimensional scanning was performed by using a microcity scanning. From the result of this 3D scanning analysis, we determined the percentage volume healed of the cranial defect, and the result shows a maximum percentage volume healed, almost 75% for the PAMAM-based scaffold, and followed by the carbon nanotube-based scaffold. The least was a macroporous scaffold, and actually the least was the SAM-based scaffold, but the SAM is without any scaffold. The mice that has not been used in any scaffold in the defective area. So the summary of the study is we have here proposed a very simple and frugal high-speed stirring-based foaming and freeze drying method, and this approach of fabrication method has successfully generated macroporous scaffold, which leads to an increased porosity and pore size and a greater degree of penetration of the bone microtissue. Now, incorporating the PAMAM dendrimer into this polymeric formulation, we found a formation of a scaffold with enhanced stiffness, and compared to the carbon nanotube-based scaffold, the PAMAM scaffold showed increased cell migration capability as well as an enhanced vasculogenic potential. This better osteoconductive and osteoinductive property of this PAMAM scaffolds results in highest healing percentage of critical bone defect compared to the other scaffold. With this, I concluded my study. These are some of the selected references. And for this study, I sincerely acknowledge my PhD supervisor, Professor Tapas Kumar Maiti, all the faculty members of biotechnology, IIT Kharagpur, the Central Research Facility of Biotechnology, IIT Kharagpur, and the UGC Fellowship, as well as IIT Kharagpur for my travel grants, all my lab seniors, colleagues, and last but not the least, a sincere respect and gratitude to my parents. With this, I conclude my study. I hope you have all liked this talk. Thank you.